Welcome to the Fourth Lane YouTube channel. Today we'll show you the celebration of 11th Foundation Day of National Institute of Biomedical Genomics NIBMG, which was held on 6th August 2020. It's a great honor that NIBMG allows us to cover and broadcast the entire event.
You're live now, Sija, start.
Good afternoon, everyone, and good morning to our distinguished speaker, Professor Sunetra Gupta. She is tuned in from the UK. Let us wish NIBMG a very happy birthday, a very happy 11th birthday. And without much further ado, let's initiate our program for today. Okay, let's start with the first thing, begin with an invocation which is recited by Ms. Shrija Mukhopadhyay. Shrija.
सर्वेषां स्वस्ते भवतु, सर्वेषां शान्तिर्भवतु, सर्वेषां पूर्णं भवतु, सर्वेषां मङ्गलं भवतु, सर्वे भवन्तु सुखिनः, सर्वे सन्तु निरामयाः, सर्वे भद्राणि पश्यन्तु, मा कश्चित् दुःखभाग् भवेत्, सर्वे भवन्तु सुखिनः, सर्वे सन्तु निरामयाः, सर्वे भद्राणि पश्यन्तु, मा कश्चित् दुःखभाग् भवेत्। शांति शांति शांति.
Thank you, Shrija, for that melodious rendition. Thank you very much. I now invite our director, Professor Soumitra Das, to say a few words on this occasion and lead us into the next program. Just please the slide.
Next up, share karo, huh? Full screen, full screen, full screen karo. Slideshow.
All right. Formally, warm welcome to all of you to this 11th Foundation Day of National Institute of Biomedical Genomics. 11 years back, little ahead of that, Parthoda, our beloved Parthoda, Professor Parthopratim Majumdar, had a nice dream of setting up one institute which will actually deal with the concept of biomedical medicine. Biomedical genomics was actually a very new concept around that time. And he conceived the idea, and the then Secretary of the Department of Biotechnology, Professor M.K. Bhan, recently we unfortunately lost him. Both of them actually somehow materialized their dream. And now this 11 years we have walked past. And now closely we are now becoming team in another 2 years. So this institute was actually established in 2009 with a vision that it will enhance the knowledge on human health and disease through genomics. The idea is to translate this knowledge using appropriate technologies for promotion of well-being and improvement of the genetics-based healthcare. Again, I am reiterating what he had a dream in 2009 was a very, very new concept. And now it is a very, very popular concept all over the world. So the institute started, and we had slowly over the years accumulated different areas, and the scientists are addressing the gene signature, the functional genomics of cancer. There are people who are working on different cancer system, like the breast cancer system, cervical cancer system, oral cancer system, pancreatic cancer system. We also have areas where people are working on the genetic determinants of the infectious disease, the susceptibility and the pathogenesis. In this also, we have people working on different infectious disease like viral diseases, sepsis. We also have parasitic disease like leishmania. We have the bacterial tuberculosis. Some people are working on this MTB as well. Then we have the strength of this institute is the statistical and the computational genomics. As you all know now, huge amount of data are generated experimentally and also in silico. How to integrate that and make a signature out of that which will have a deliverable is the area of this statistical and computational genomics, and we are actually addressing in different facets of that. And finally, we also have the multi-omics determinations of the complex disorders. This is the little disordered sector because we have different disorders we are addressing, starting from liver disorder, fatty liver disorder. We have also ocular diseases, we have other areas, and people are actively participating. Over these years, this long journey, the Institute has actually set up a lot of collaborations, and Institute actually championed in coordinating multi-institutional projects. I mean, National Institute of Biomedical Genomics is one such example of this Indian institute who can actively coordinate with different institutes. And one such example is the SciMac project, which we call the System Medicine Cluster. It comprises of 6 different institutes, as you see they are listed. NIBMG is the central. We have the ISI, Indian Statistical Institute. We have the Indian Institute of Chemical Biology. We have Bose Institute. We have ISAR Kolkata. All these institutes are actually addressing different infrastructure they have set it up and different aspects of 2 different cancers, mostly the oral cancer and the cervical cancer. The idea is to provide improved tools for prediction, prevention, and treatment of disease using system biology approach. And the idea is also to create an infrastructure in Calcutta. It's often called as a Kolani cluster. Create a platform for a multidisciplinary training to build a cadre of scientific, clinical, and technical personnel required to drive and sustain system medicine. Again, this is the brainchild of our beloved Parthoda. Now, Along with that, we also have an International Cancer Genome Consortium, ICGC, where also you can see that NIBMG has played a major role from India to obtain a comprehensive description of the genomic, transcriptomic, and epigenomic changes in 50 different tumor types, subtypes, and which are of clinical and social importance across the globe. And to facilitate this, Government of India has now given charge to National Institute of Biomedical Genomics with this coordination from Professor Partha Majumdar to set up a national genomics core. The idea is to provide a high throughput platform facilities and expertise for generations of genomic scale data, provide facilities and expertise for big data storage, management, access, and analysis, make the genomics facilities available to many sectors, including basic biology, health, agriculture, forestry, livestock, marines, etc. Along with NIBMG, we also have CDFD, that Center for DNA Fingerprinting at Hyderabad, and we also have an University of in Allahabad, another centers which are coordinating along with this National Genomics Core. NIBMG is also taking part with the Grand Challenge program of the preterm birth. As you know, that almost like 8,000 pregnant women are being now screened for the signatures for addressing whether there is any reason to get into a preterm birth or even there are other problems associated with the pregnancy. The way we are studying, this is actually along with the three other national institute, THSTI, which is in Faridabad, and RCB, both of them are Faridabad, and there are several hospitals along with them. The idea is to find out the epigenomics, genomics, and their contribution, and how the stress, the development of methods to predict preterm birth based on the genomic correlates of the stress. This is again a challenging area, an upcoming area, and NIBMG is championing in that sector as well. Now, another initiative the Government of India has taken is the Genome India. As you know, India with a diversity of 1.3 billion people and 4,000 caste and subcaste, 4 languages families, 461 languages and 26 scripts. So this is a unique situation where people can actually find out the diversity and try to find out the meaning out of that. They are trying to address it with 52 different centers, and NIBMG is part of them. NIBMG is one of the 4 institutes who will collect samples, stores, sequence, and analyze. Is there a problem? What happened? Okay. Yeah, so this things is addressing the global problem of Eurocentricity in the studies. The idea is to catalog the human genomic variations and spatial reference to India and reference databases to design and conceive human genome studies of the Indian population, understanding the historic and the prehistoric demographic events, a resource for reliable imputation of the haplotypes estimation in studies conducted on populations in India, and finally design population-specific genotype chip and biobank. This is an unique opportunity to contribute to the society, and then again, NIBMG come forward and contributing heavily to this initiative. We recently have a scientific advisory committee meeting, and NIBMG's role was highly appreciated into that. And you know, all this started again in a small way, because we have established, again, Parthoda has established a Kullani cohort. So there are people, this is a peri-urban area where almost 20,000 individuals were actually identified from villages in and around Kalyani, and then try to see the prospective studies on the genomics of health and disease. And then we are trying to understand that is that a possibility that if we can track over the years, and can we really understand how the optimal lifestyle strategies can divert or perturb. And we are trying to somehow use this cohort for another flagship project of our institute on the cervical cancer, where HPV screening program and the Genome India project, both of them are actually taking help from the Kalyani cohort. Institute also serves to the society by one genomic service unit that is actually, initially it is housed in the PG Polyclinic in Kolkata, but we are trying to start something in Nadia district as well. As a part of the program of Government of India called UMEED program, we are trying to apply for that. This genetic service unit actually has standardized whole bunch of genetic disorders to be detected, and we want to now translate this into an application mode and try to screen the high-risk pregnant mother and the newborns and try to understand whether we can actually help them in predictions. And as I just briefly mentioned, the flagship project is again how to integrate the multi-omics data using big data analysis to understand the optimal life course trajectories and a better management for non-communicable disease. We have taken cervical cancer as an exemplar, and we are actually using Kalyani cohort to start with, and we are also ties up with two other hospitals. One is TMC in Kolkata, one is JNM in Kalyani. Now, finally, I would just try to draw your attention that NIBMG actually coordinated a program called Pan India 1000 Search RNA Genome Sequencing, and it is successfully completed last week. And then what we actually try to do is the sequence of this, and then try to now analyze this sequence and try to find out how the phylogeny of the virus, which will indicate how the track of how the virus is evolving in different states within the India. This is actually across 14 different states, and we have collected samples from all over. You can see there are 5 different DBT institutes involved into this, but they have taken help from different research institutes and hospitals across India. As you see, those yellow spots actually indicates different hospitals and their locations. We collected samples, sequenced it. Again, the sequencing is done at the unique facility of NIBMG, that National Genomics Core of Pattoda, and actually we could now try to analyze and make some meaningful deliverable from this. This is essentially the imaginations of our academic coordinator Alpana, and she feels like this is the way we can actually reach out to the people, that what is the outcome of the sequence analysis. But actually, to tell you the truth, we are still analyzing and try to go deep into it and try to understand whether this sequence information will be very, very useful for the predictions in the years to come. Now, I am actually missing all of you in the campus. Traditionally, we meet in our seminar hall, and I was just hoping, I wrote a one-liner to Professor Gupta that we wish that we could have you here, and then we could show you around the campus and interact with you more. Our students are actually our assets, and as you see, they are all actually taking parts in different activities around the year in the institute. As I said, I begin in the beginning that this is that 11th year of the institute, and we are actually growing, and this is a very, very crucial time that any decisions, any initiative we take, we should think twice so that actually this will be really shape up our future. And with all these things, and again, on my personal behalf, actually, when I mentioned we had a meeting with the Secretary of the Department of Biotechnology today, and when I mentioned all the directors, including the Secretary, actually congratulated National Institute of Biomedical Genomics on this birthday. And I would like to congratulate all of you, and again, welcome to this meeting. And now I will over to Pattoda, and then he will give his remarks and also introduce the speaker. Thank you.
So it's really my privilege to introduce our Foundation Day speaker this afternoon, Professor Sunitra Gupta. We are actually honored to have Professor Sunitra Gupta deliver the 11th Foundation Day lecture of our institute. Of course, you know that Professor Gupta is a professor of theoretical epidemiology at the University of Oxford. She runs a wet lab and does profound mathematical modeling influenza, meningitis, etc. She is now involved in various aspects of SARS-CoV-2 infection. Of course, you may also know that Sunetra received the Rosalind Franklin Award of the Royal Society for her scientific achievements. But how many of you know that she's also received the Sahitya Academy Award from the government of India for her novel that was published in 1996 called Memories of Rain? How many of you know that she has translated into English many poems of Rabindranath Tagore? Who, she felt, is perched upon a cloud and gently drops sheets of poetry onto this earth. Sunetra does not see division between arts and science, only different ways to express ideas. Sunetra was born in Calcutta. After spending her childhood in Ethiopia, Zambia, and England, she returned to Kolkata at the age of 11. She received a fellowship and moved on to Princeton to study physics. In addition to courses in physics, she took a course on animal behavior. And I quote from her, "I was fascinated to see that mathematics could be used to make sense of biology." She then went on to take a course on theoretical ecology that also included discussions on infectious disease epidemiology given by none other than the legendary Robert May. She has acknowledged that this course and the influence of Robert May has had a major impact on her career. She decided that that is exactly what she wanted to do for the rest of her life. Sunetra got her PhD from the University of London in 1992 on modeling and epidemiology of infectious disease. With a grant from the Royal Society, Sunetra has collaborated with the illustrator Ted Duan to uphold remarkable women scientists such as Barbara McClintock in a project titled The Shooting Stars. She is now writing a book on these remarkable women scientists. I could go on and on describing the diversity of Sunetra's contributions and achievements, but I'll restrain myself. I've written that SARS-CoV-2 coronavirus is very kind, but today I feel that the virus has been very cruel to all of us. Wouldn't it have been awesome if Sunetra was standing in our lecture hall and delivering our Foundation Day lecture? I also had a secret wish. I wanted to get her autograph on a copy of my copy of the book that she has written, So Good in Black. All these wishes have been shattered by SARS-CoV-2. We now have to listen to her over the web. Models and Metaphors, Professor Sunetra Gupta.
Kartiki, now you take a call.
Hi, Vikram, can you take over and we'll have Professor?
Yeah, I'm doing it. Ma'am, are you ready?
Yes, I'm ready.
Yeah. Okay. Could you please share your screen now? Or
I'm going to turn off my video just so the screen says the network quality is not great.
yeah, it is coming.
Can you see it?
But you put up your camera?
Shall I put my camera back on?
Yeah. Because you have to be visible.
Well, I'd like to be visible.
Yeah. Yeah.
Should I start?
Now we can start, ma'am. Now we can start.
Okay. Well, this is really quite emotional for me because it seems so strange to be sitting here staring at my screen instead of being in Kolkata. It's, it's just beyond tragic. I wish I were there, and all I can say is I will be there at the earliest opportunity, and I would love to meet all of you face to face and to see Bhartada again. Thank you so much for that very fulsome introduction. It was so nice also to have that invocation and also to hear a little bit about all the extraordinary work that's been going on in your institute. And as I said, I really hope that the situation soon permits me to come and visit you all. So I agreed that I would talk today about models as metaphors, and as I was putting the talk together, I thought maybe because models in some senses are perforce metaphors, maybe just to alter the title a little bit to models and metaphors, one being a reflection of the other. And I'm going to start, I think, with models as applied to science, because as Bartha said, that's what I decided some, God knows, 35 years ago now, that would be the substance of what I did for my as part of my academic career. And indeed, what I have done, I think, over the last since then, is try and fulfill that goal of using models to try and understand biological systems. And of course, in that process, I've learnt something about what models actually can deliver. And I hope that I can perhaps transmit some of that to you in the next, or during the course of this talk. So, one of the most important messages I try and get across, both to my students and to the general public more recently in the context particularly of this pandemic that is now causing me to be sitting staring at my screen instead of your faces, is that, I mean, in this pandemic, mathematical models seem to have played rather a large role in our understanding of how it's progressing, but also in terms of informing policy. And one of the things that I think people need, so suddenly mathematical modeling has become something that everyone feels they either do understand or need to understand. And I think the central feature of mathematical modeling, central kind of tenet, which I think, in fact, not just the general public, but even practitioners of mathematical modeling tend to sometimes not entirely realize, is that mathematical models are abstractions and not replicas. So here I illustrate this often by means of this analogy, where you could have a toy car. Here are two examples of what a toy car can be. And one of them, as you can see, is very detailed. And if you didn't have the reference of the hand that's holding it, the toy car on the right could easily be seen. You might mistake it for a real car. So it's very detailed. Whereas the one on the left is clearly an abstraction. It doesn't resemble a car in any way, shape, or form. And yet, you can imagine that if the sort of very simple wooden little toy, if that had wheels that moved, and if the replica didn't have wheels that moved, that in fact the better model of a car would be this very abstract wooden toy on the left. In other words, a child playing with this simple Brio car would learn a lot more about how cars moved than from this other sort of replica, as it were, particularly if its wheels didn't move. So that's kind of a very simple way that I make this point, that mathematical models are abstractions and not replicas, and indeed simple mathematical models often have more to deliver than very detailed and complex models. Now, so if you think about this sort of axis of going from a simple model to a complex model, one of the dangers of having a very complex model is not simply just that it has less to offer in terms of the connections between pattern and process, but that indeed it can sometimes seduce the user and the person who is trying to assess the results into, it can seduce the person by its very sort of appearance of being closer to reality. And a concept that I, again, have used to illustrate this problem of achieving or moving towards something that is apparently more realistic, but actually doesn't illuminate the system or give us more of a sense of the dynamics of the system that we're trying to understand. One way of approaching that whole problem is with reference to this concept of the uncanny valley, which was introduced in the 1970s by the robotics professor Masahiro Mori. And what he pointed out was that if you, in the field of robotics, if you go for, obviously people try and make robots look more and more human. And what's interesting is as you go from a completely machine-like entity to something which, if you increase the anthropomorphism of it, if it starts to look more and more human, indeed, to start with, your emotional response increases. You become more, you develop more empathy towards this robot, which, as it starts to look more and more human. But then there comes a point where actually before it starts to look fully human, at which point obviously you respond to it as a human, there is what's known as the uncanny valley, where the robot looks very, very human, but not quite human enough. And what that generally elicits within us is a feeling of fear and disgust. So this uncanny valley is sort of shows this, if you like, a non-linear relationship between achieving reality and, in this case, of course, the emotional response. And one can use this kind of conceit to explain why complex mathematical models also can fall into this trap, in that if you substitute the attractiveness, the y-axis for, if you like, the utility of a mathematical model, and what I just described to you was a scenario where a mathematical model might be very, very simple, or any kind of model, a toy, if you like, be very simple, but have components that allow someone to make the link between the movement of the wheel and the direction that the car goes in, as opposed to a car that is much more realistic, but may not have wheels that move, which may look more like the real thing, but in fact, in terms of its utility, is actually experiencing a dip, in that in trying to become more real, it is losing its value. So that's the kind of general framework, my sort of general philosophy of modeling, which I just thought I would get across. And what I thought I would do next is to try then to see how the kind of modeling exercises that we've been performing ourselves and other groups, how they fit into this scheme. So this is already out of date, of course. This is data on deaths associated with COVID, SARS-CoV-2 in the UK, and there is some discrepancy depending on, I obviously don't have time to go into all the problems and caveats with data collection, but overall what we saw in this country with lockdown being introduced towards the end of March is that there was a peak of deaths around very early in April, and then a death started to come down. And indeed, since lockdown has been lifted, there have been a few outbreaks here and there, but certainly in places like London, it seems that whatever happened during that peak has been sufficient to prevent it from coming up so far. Now, obviously, one can't, once lockdown happens, you need a model that incorporates lockdown in order to try and make sense of the data. But if you look at the data up to the time of lockdown, it is possible to try and fit a mathematical model that incorporates the processes of the spread of the epidemic in order to try and make an assessment of where it would go if one were not to lock down, or indeed if one were to lock down partially, or what to do now with the lifting of lockdown. And so what we did at the time was to put out the results of a very simple mathematical model, one that would be the equivalent, if you like, of the little wooden toy car. It's a very straightforward model, compartmental model, which divides the host population, in other words, humans, in this case, into those who are susceptible, those who are infected, and those who have recovered and are, for the time being anyway, immune. And so within this kind of structure, a very simple structure, you can try and understand how the epidemic will spread by allowing people to flow from the susceptible compartment into the infectious compartment at a rate which is determined by the per capita risk of infection. And then you allow people to move from this compartment into the recovered compartment at a rate of recovery, which is fairly one parameter that is in fact not that difficult to measure. And effectively, when you take this model and you write down a set of equations that captures what I've just described, that structure, then you can see, and this is not, this is sort of fundamental epidemiology. It's not sort of our model in any sense. What you see is if you start off with a population where everyone is susceptible, so that blue line measures who's susceptible, and no one is immune to SARS-CoV-2 at that point, what you will get is you'll get this growth in the numbers infected, shown in red, and meanwhile numbers susceptible naturally will start to fall. And there comes a point, a critical point, where the numbers infected starts to drop. And this is because the proportion of the population that is already immune at this point stops the numbers infected from growing. They will still continue to increase, but their rate of growth will become negative at this point, which we term the threshold of herd immunity. That is to say, a point at which sufficient people are immune that the epidemic will naturally, at this point, die out, even if no further people were infected. Of course, what happens in real life is more people get infected, and so one overshoots that threshold. But once you've reached that threshold, the epidemic will effectively be under natural control. One of the important determinants of how quickly the epidemic will grow and what level of herd immunity is required to keep it under control is a concept known as the R0 of the pathogen, which is its fundamental transmission potential. It has a simple biological interpretation, which is that it is the number of infections that would be generated by a single person, infected person, in a totally susceptible population. And it can be expressed as a combination of parameters related to transmission as well as the duration, the rate of recovery. So it's a combination of parameters. Obviously, we don't need to go into that in any detail, but as such, what it is a fundamental measure of its transmission potential. And so with this model, one can, of course, qualitatively, you can see that it matches what happens. You see an epidemic increase, you see the numbers of people who are susceptible declining, and the numbers of people who are immune increasing. Now, you can do this using a much more complicated model, which is what our colleagues at Imperial College did. So they built a computational model which explicitly mapped how the disease would spread on a spatial scale within the British Isles. So within that kind of an exercise, you don't have a set of equations per se. You have, it's a computer model, so you update the nodes of that model by a set of rules, but those rules effectively capture exactly what's going on in the simple model, SIR model I showed you last time. So effectively the same kind of process is recapitulated by this rather more complex model. These are actually figures that I've taken from a model they did for the spread of flu about 15 years ago, but fundamentally that's the kind of structure they used to look at the models that how SARS would spread, SARS-CoV-2 would spread, COVID would spread. So this model is obviously a lot more complicated than the simple SIR model. So therefore I've represented it here with something a little bit more realistic, if you like. But one of the things that I would like to stress is that I personally don't think that that kind of exercise as such offers very much more in terms of utility than the simple SIR model. So if we think of it as it may represent an increase in complexity, but as far as utility goes, it does the same thing. It can fit the data on the rise in infections and try and make inferences from that about where the epidemic will go. Now, the crucial parameter in figuring out what will happen with this epidemic is not just whether the R0, if you like, or how quickly it will grow, but crucially, how many people are likely to die upon being infected. So depending on whether you think that the infection fatality rate is around 1% or around closer to 0.1%, you can get radically different figures for the cumulative mortality using this model, whether you use the big, really simple model that we showed or the much more complicated computational model of the Imperial, that the Imperial College people used. So then the question, of course, is, well, which fits the data? Which of these models, which of these parameters actually fits the data? And the thing that we tried to point out in March is that they all do. That at that point, pre-lockdown, the growth that we had in cumulative numbers of deaths, which is the only thing one had any form of reliable information on. Cases, as you know, depends on how many people tested, where they're tested, what tests are used. You know, it's not something you would, it would take a lot of sophisticated work to translate cases into the real numbers in order to fit the model. But we can fit the model to the cumulative deaths. And the truth is, any of these models will fit what happened in this country up to the point of lockdown. So our point in March was simply to say that the Imperial model, which also of course fits this data, is a worst case scenario model in which they have assumed a very high IFR, which at the time they did based on the data they had from Diamond Princess and a few other locations in Wuhan and elsewhere. And they, on the basis of that, they chose a very high IFR, which gives you this very high cumulative mortality. So that's the cumulative mortality, and this is mortality in time, cases in time. And what we showed is that you can equally well fit this part of the curve to a scenario where the death rate is much, much smaller. The cumulative death rate is going to be much smaller. And all that entails is that the epidemic would have started earlier. So in other words, you've got these two extreme scenarios. One where the epidemic has only just started and it's killing a lot of people, and therefore that's what you're seeing in this rise of deaths. And the other option is one where the epidemic came in some time ago. When I say some time ago, I mean January. So if you have an introduction date that is close, so introduction date meaning when the epidemic actually started to take off, it may well have arrived earlier, but not properly taken off in the communities. If you have an introduction date of around in late January, you can easily get the same rise in deaths, but for a much lower infection fatality rate. But what that would mean is that a lot of people would have already been exposed, and the signature of this should therefore be evident in any serological survey that we do. So at that point, we set out to try and do those serological surveys, and Craig Thompson in my research group immediately, with incredible speed, developed a neutralizing antibody assay, a pseudotype neutralizing antibody assay, which we have since then been using to try and understand what the pattern of what the level of seroprevalence is in the population, as an indication of how many people have already been exposed, in order to understand what the real death toll would be in the absence of lockdown, since now, especially, we were aware at that time that lockdown could not be a long-term solution to the problem. As Bartosz recently said in an article that he very kindly sent me, it was perhaps necessary to impose lockdown at that point simply because we didn't know what the nature of the beast was fully. But now we do have an idea, and now lockdown is no longer a viable option unless we're happy to let lots of people starve and die of other preventable diseases. So now we really need to understand what proportion are actually immune. Have we hit herd immunity? Where are we going with this? What are the reasonable options? So at the time, we thought what was the answer to that question would be fully met by determining the levels of exposure in the community. And so we set about trying to, it was very difficult to get the necessary samples to understand what was going on because of the huge amount of politics and both the government level of the government and not to mention in academia itself. And eventually the Scottish Blood Donor Service gave us some data, and this is just a preliminary analysis of some data from March, where we found that between the second week of March, third week of March, we could see cases starting to grow in parts of Scotland, but other parts of Scotland, there were very, very few seropositives, and that we've now analysed the data all the way through till May, and it remains the case that some parts of Scotland seem to have very low seroprevalence, whereas there are areas in Glasgow where we've recorded a seroprevalence of 10 to about 10%. The figures that are coming out of somewhere like Dharavi, for example, or indeed today, the Afghanis have reported that a third of the population appear to have been exposed already. Infection prevalence in New York, we've seen sort of average seroprevalence of about 20, but there are areas of Brooklyn and the Bronx where it can be as high as 60%. These are data that were presented on people coming in, women coming in for delivery, and showing what the infection prevalence was in these women. And as you can see, it was pretty high in the Bronx, but overall, it was, you would say from these data that there was a lot of exposure occurring in New York in March and April of this year. And this perhaps can explain why now we aren't seeing the kind of resurgence that you might expect with lockdown measures being lifted. But so the fundamental point there was that as data accumulate, we can perhaps start to discriminate between these scenarios where you have an extremely high infection fatality rate and as opposed to one where the infection fatality rate is much lower. And that is really a very crucial element that we must absolutely need to get a handle on in order to figure out where we're going with this epidemic. And of course, what we know now is that the vulnerability of the population to death, severe disease and death, is not uniformly distributed, that these deaths are confined to the frail and the elderly and those with comorbidities. And so it is not surprising that in countries that have a different type of age structure, which is not so heavily weighted towards the elderly, that the infection fatality rates are considerably lower than those that have been observed, for example, in the province of Bergamo in Italy, where it took such a toll. So just to go on from that idea that, actually, one of the things we have very, we've learned that we can be confident of, now that the epidemic, we've watched this unfold, is that vulnerability is not uniformly distributed. And so what we've moved on to doing is looking at models where this is the case. So you have, instead of just one population characterized by a single SIR, you have a population, a general population that has, that you characterize, where you have this SIR dynamic occurring. But this is linked to another population, a subset of the population, which is either more vulnerable to death and disease, or, as we are starting to now realize, and this is really crucial, that there is also a subset of the population that is completely resistant to disease and death, and indeed even infection. This was not something I expected when I started to work, when this virus appeared. I had thought perhaps that previous exposure to other coronaviruses might stop severe disease and death, but it's data now emerging that it actually stops infection itself, which is, to me, quite surprising, but really actually fantastic news. So once you have that kind of a stratification, so now you're making a model a little bit more complicated. So we've still got a toy car, but now the toy car looks a little bit more sophisticated. And in this one, you can sit and spin around. I have very fond memories, actually, when I visited Kolkata. I was 4 years old. One of my cousins had a car like that, which spin around the veranda on it. And anyway, just a digression there. So I've always coveted one of these. Anyway, but once you have two different groups, and you're looking at, so you've elaborated your model, you can see that a number of different dynamics are possible. One, where the vulnerable people become infected first, and so you might record the deaths in the vulnerable population, and get from that an impression that the epidemic is spreading very quickly, and also that the death rate is very high. Whereas actually what hasn't happened yet is that it hasn't reached the general population, which it will later, in which case you'll get a later peak in the general population, which will not be carried the same level of have the same death toll. Alternatively, you could have a scenario where the general population has already been infected and it's peaked and it's on its way out, but it's only just reached the vulnerable people, in which case you see the deaths happening later, as we did here in many of the care homes. So deaths sort of happened much later. So that's if you stratify the population according to who's vulnerable to disease. But what, as I said, so that's crucial in the design of policy, because I think one of the things that we really must consider is how we can shield the vulnerables in the times ahead, because obviously going into lockdown to shield the vulnerables, I think, is not really an option anymore. But what is now actually turning out to be another dimension to this whole pattern is, again, a question of difference in vulnerability, but this time what we're now starting to see from a whole set of studies is that some people are actually resistant to infection due to, quite likely due to having T-cell responses to other circulating coronaviruses. So this graph here from this new paper published a few days ago, last week, shows that the sequence identity between different targets of T-cell responses in different parts of the virus between SARS-CoV-2 and these 4 circulating endemic coronaviruses. And that seems that is high, and also we now know there are immune responses in people to these viruses, and that there is a correlation between having these immune responses and being protected from infection. So these people aren't developing antibodies, which is a bit of a blow for those of us who were hoping to use antibody serosurveys to assess exposure. But in other ways, it's extremely good news. And so we have just put out on medRxiv a paper which, again, takes into account this feature that a proportion of the population might be resistant, and calculates how the herd immunity threshold under different levels, types of mixing between the resistant population and the general vulnerable population, how that can alter the herd immunity threshold. Now, you'll see reports everywhere of herd immunity having to be around 60-70%, and that is based on the idea that the R0 of the pathogen is essentially around, very much, certainly in excess of 2. R0 of 2 would give you a herd immunity threshold of 50% by a very, very simple formula. But what you can see here is that if there is the proportion of people resistant in the population increases from, even if you've got 20% of the population that is resistant to the infection, then this threshold can come right down. So this is what happens under assortative mixing, that is to say, if resistant people mix only with resistants and susceptibles only with susceptibles, but that's very unlikely. This is the more realistic scenario where they mix, because you don't know who's resistant and who's susceptible. So we're all mixing in a random way. And what you can see here is if R0 is 2 and the proportion resistant is 0.3, you're really already in a territory where having 10-15% of the population being seropositive is already guaranteeing herd immunity. So this is good news. We need to, obviously, we don't know what proportion are resistant, although some papers like this one that I've put out there suggest it might be 30-50%. So we could, in many areas, actually have already approached and overshot, in fact, that threshold for herd immunity. And this framework also helps explain why in some areas you see very, very low levels of seroprevalence, even though the epidemic has, certainly the virus has spread in that community. And then, of course, why in some communities it's hardly taken off at all. And that could do with the proportion of the population that is already resistant. So overall, what that shows is that these exercises illustrate is that adding some complexity can certainly increase utility of a mathematical model, but one has to still be very, very careful to then not add further realism that actually just brings you back down to where you were before, or indeed could even drop you further down to where it becomes less useful. So a really complicated mathematical model with the kitchen sink thrown into it is really useless because you can't use it to infer anything. So in terms of, what should we then be aiming for? Where should we go? Where should we position ourselves in this sort of scale of complexity? I think this article from Tim Newman is really quite useful, which says that simplicity has to be our ultimate ambition in science. Simplicity is power. Simplicity in its purest form takes a small number of inputs and gives a large number of outputs. These outputs are generally assumptions, and the inputs are generally assumptions. The outputs are generally new concepts and predictions. And it's the ratio of these that actually we should watch out for in conducting this sort of exercise. And as he says, theories with an output-input ratio less than 1 should be discarded. Such theories have been described delightfully as not even wrong, which apparently Pauli said of such models. So we do want to stop descending. We have to be very careful to not be seduced by this idea that the more realistic things are, the more valuable they are, because what that can lead to is this distortion of the input-output ratio to the point where you are led into the uncanny valley. So now I'm going to divert from that. I'm going to make a link from this concept to the other side of what I do, which is write novels and translate poetry, and indeed read novels and read poetry, which is something I like to spend the better part of my day doing when I have the chance. And so linking to what I just said is this lovely quote from Georgia O'Keeffe, where she says, "Nothing is less real than realism. Details are confusing. It is only by selection, by elimination, by emphasis that we get at the real meaning of things." And of course, she's not talking about mathematical models. She's talking about her own art and the art of other people who, again, have often eschewed realism in favor of abstraction or surrealism or some other form, which is not realistic. And this is her reason, her explanation for why we might move away from realism. And again, I mean, there's a real kind of resonance there with what we're taught often in school, which is that art is best when, you look at painting and say, oh my God, it looks great because it's really looks exactly like the real thing. Here's a flower, it looks exactly like the real thing. Or he says, Chandra Chattopadhyay is fantastic the way he depicts the real life of the village. And of course, both are all true and have value, but there's also this other way of approaching the depiction of reality, which is not actually one that adheres to a faithful replica of reality. So sometimes the better way to get at reality is not through replica, it's by use of abstractions. And that is indeed the guiding principle in how I write. I steered away from realism from probably from a very young age. I mean, I grew up in Kolkata, and major influence in my life, the most important person in my intellectual development was my father, Thrugugupta, and he was an art critic, a film critic. He wrote himself, he did everything. He also, of course, taught at the university. His day job was to teach modern history, African history in particular, but he was very active in the arts and introduced me from a very early age to this concept that reality doesn't have to be approached through realism, and it is very much to him that I owe, funnily enough, not just my literary endeavors, but I think my understanding of science also derives from those crucial lessons that he delivered to me on how to approach reality and try and make sense of reality. And so, as you can see, this is just how I start my last published novel. It's been a long time since I wrote this book. It actually first came out in 2009, which is an important date clearly for your institute as well as myself. So this was published in 2009 and in India by Women Unlimited, Ritu Menon. And I will very, very gladly sign the copy for you both when I'm back in Kolkata, or if we meet here. And this is how it starts. This is a scene in Digha, by the way, in my head. Child on the seashore, I loved your mother once. How unkindly these words pound through my blood as I walk down this dry path towards you. How cruelly they beat their rhythm within me, as though already relentlessly rehearsed. I halt at the low iron gate that separates the garden from the beach. From here I have a good view of you, a slender young thing in a crimson frock. You stand with your feet planted slightly apart, turning your head slowly from side to side, methodically surveying the various life forms deposited about you by the retreating sea. Those that you wish to save, you seem to be trapping under glass jars, into each of which, when uprighted with the sea creature squirming within, you ladle a liquid from a bucket by your side. Seawater, perhaps, or some other kind of nourishing brine. A dog barks, causes you to look briefly over your shoulder. I catch a glimpse of your delicate features, your large, quiet eyes. My head reels. Again, I feel a sickening familiarity with this precise moment, as if it might somehow already be embossed upon the meager felt of my imagination, a moment such as this. I went to Digha again on one of my holidays. My father's uncle lived there, and I have very lovely memories of being on the beach there and indeed catching something, a crab or something in a little jar. So that's where this partly comes from. But of course, I've chosen for it to be told not by me as the child on the seashore, but through the eyes of an American journalist. So again, placing myself in looking at this scene through somebody else's eyes entirely. And also you'll notice from this that the way that I use grammar and language and words is not out of a consideration of realism. And indeed, one of the things I mention in terms of the difference, however, in what I aim for when I'm writing as opposed to in science, which I hopefully have illustrated and goes back to that Tim Newman, what I read out, in that there I'm always looking for simplicity. Whether it's in writing the equations or constructing the model or interpreting the results, I'm always looking for simplicity. I'm always looking a gap as possible between the word and what it means, what it refers to, the word and the referent. Whereas in this, as you can see, the word, I'm almost exploiting the gap between the word and what it might possibly mean. So in literature, it's the relationship between reality and representation of it, and the understanding of that reality derives from the gaps that exist between the word and its referent. And also in terms of grammar, what I realized, I started to write and I didn't know what I would do with grammar. I used to write in Bengali, in Bangla when I lived in Kolkata, but when I started writing again in the UK, while I was doing my PhD, because I felt it easier to write in English. I didn't know what I would do with grammar, but as you can see from here, again, I found myself leaning towards an unconventional use of grammar. And one of the things I realized is that my relationship with the punctuation itself was really quite visceral. There's something that it's a felt kind of relationship. And I find it hard, even though here I have used some full stops. I don't like using full stops. There's something very final. It's like a bullet into the page. Whereas, in fact, our dari in Bengali, I think, is a much more gentle way of ending sentences. So I realized that I was responding to the text in a sort of visceral way when it came to punctuation. But then I found several examples of this in other people's texts. So, for example, one of my favorite writers is Virginia Woolf. And the funny thing here is she uses semicolons. You see this in this paragraph. She litters her text with semicolons. She uses them to such great effect, whereas I can't use semicolons when I'm writing literature. I like to use them in science, but not when I'm writing literature. And then, different people have different things to say about semicolons. A wonderful poet, Arvind Mehrotra, in one of his essays, mentioned that the semicolon, he was writing about somebody else's work. The semicolon in the first sentence is like an osmotic membrane. And then Kurt Vonnegut, in his typical florid way, talks about them as transvestite hermaphrodites representing absolutely nothing. So people can become very, very emotional, have an emotional response to punctuation. And this is just another, just in the interest of time, I won't go. Emily Dickinson, another favorite poet, uses dashes through her poetry, some of which was sanitized when they came to be published, but now have been reinstated in most recent publications. And there's some lovely interpretations of what these dashes mean. Brenda Hillman talks about them creating the sensation of walking across a rope bridge, strung across a raging river below. Susan Howe talks about them as stitches through her poems. Lyndall Gordon says they push the language apart, creating spaces for the reader to fill. There's lots of ways in which you can interpret these punctuation devices, which show you how their unconventional use can be really productive. These are just some other examples, and in the interest of time, again, I won't go into them. I talked about the full stop myself that seemed like a bullet, and John Banville says that it's like it's been stabbed onto the death certificate. But Banville also speaks of tenses in physical term. I cling to the present tense as to the Shearcliff's last handhold. So we've gone from word to punctuation, and finally, narrative itself. How does one construct narrative? And this is where I think actually, I've talked about differences between science and literature and the use of words and punctuation, whereas in science I like to use and adhere to a very strict kind of regime and to make it as simple as possible, as clear as possible, whereas in literature it's sort of the muddle that makes it exciting. But when it comes to creating narrative, I think both science and literature engage, for me anyway, it occurs very much in the same way. And this is a sort of a box, a metaphor, if you like, I found for narrative itself. It's been removed from the National Gallery here in London, but it dates back from the 16th century. And it's when people were really obsessed with perspective. And this box is painted on 5 sides in a way that if you look from the 6th side, just looks like almost like a cubist painting. It's all a jumble. But then there are these holes drilled into either side. And if you look through these perspective holes, the scene inside arranges itself into this, really rich, delicate and beautifully proportioned interior of a Dutch house. And I feel that the way that narratives are constructed in science and in literature actually conform to this principle, whereby you often have this jumble of, or a sort of set of jigsaw pieces, a jumble of facts, observations, and our role as scientists and as literateurs is really to try and construct a coherent kind of framework, to find a way to drill the hole in the right place, to put this all together, to say, oh, look, this is perhaps what is going on. This is one way of trying to create order from the disorder that we're presented with. But we must be careful in that to not sort of shoehorn it into some sort of linear form. Again, to avoid the sort of compulsion to make it, to move towards a sort of form of realism with a beginning, middle, and an end, something that's easy to sell, if nothing else. So we have to make sure that we create order from disorder, but not shoehorn it too much into a sort of linear narrative. So indeed, when it comes to complexities of narrative form, we must remember that the linear narrative is really only one very simple option, and science itself suggests that there are other forms which can be very complex and yet follow some very simple rules, such as the fractal form, and which all speak to the fact that art, to use a beautiful quote from Lyndall Gordon, art is made at the interface between abandon and decorum. And in some senses, so is science. It's letting the imagination unfold, but also containing it using these wonderful methods that we have at our disposal, of which mathematical modeling is only one example. So I think I would like to end there. I just wanted to say a few words about which a topic that I've spoken about several times now in two conferences, both organized by Amit Chaudhry. The first was on deprofessionalization, and more recently, last time I was in India in February, was on the concepts of failure. And in both cases, I found myself speaking about the condition of being both a scientist and a writer at the same time, which is something that is often not looked upon as a sort of sensible way to be, both by scientists, but more often actually from my literary friends, often sort of ask me, well, why are you doing the science? And the truth is that the reason, their rationale is obviously if you do one thing, you might do it better by just sticking to that one thing than doing two things. And my answer to that is really that I'm not trying to do something, I don't do it because in order to be the best at it or to do it to the, I do it as best I can, but I think that for me, the idea of being in a profession is not one of being successful and trying to, compete with everyone and emerge as having made the best mathematical model or written the best novel. In some ways, being in any profession, a profession is really a space in which all we can do is try. It's a kind of permission to fail from society. It's a permission to keep trying and keep failing. And I think that it's very difficult for me anyway, this quote from Holden, which I won't read out loud because I'm running out of time. But, I think that one has to be true to oneself. So for me to give one up and do the other would be not being true to myself. And I just end with a quote from Mark Rothko, which I think kind of brings everything together, in which he says, I'm not an abstract painter. I'm not interested in the relationship between form and color. The only thing I care about is the expression of man's basic emotions: tragedy, ecstasy, destiny. Thank you very much for this opportunity to speak to you. And as I said, I hope I will be able to meet you all face to face. Sorry, I'm trying to do it.
Kartiki, wait.
Is that all right if I stopped?
Oh, that
Yeah.
was that. Yes, Netra, that was absolutely stunning. And like I said, I don't know if you heard me, at least me and for many of us, we go back with the message that it's all simplicity. Modeling is one of simplicity. We must adhere to the simplicity of a mathematical model in order to explain reality. That's the take-home message for me, and I suppose for many of us. On behalf of the entire institute, let me thank you for the fantastic talk that you gave, connecting science and ending with literature and with Mark Rothko's motivation from also I actually remember my visit of Chapter, which is kind of gloomy. It's all black and white, but yeah, you did make a point with station. We look forward to your visit to our institute, and that's something that we will demand of you when you on your next visit to Kolkata. And again, thank you very much. I know that you have come back from a long journey, and you take care of your lab and your team. I wish you all the best, and thank you again for this wonderful Foundation Day lecture that you have provided to us. Thank you very much, Netra.
Thank you so much, and very good luck to everybody, and hope to see you soon. Bye for now. Bye-bye.
Bye.
Thank you very much. Thank you, sir, for introducing and thanking the speaker. Now we will move on to the second half of our program today. Here, what we're going to do, we know that our young NIBMG stands very tall on shoulders that are equally young, and those are our young investigators. So this year, we will showcase 3 areas that NIBMG is shaping up to be a world leader in. And we have 3 young speakers that will discuss with us their experiences and their setup and their results in the form of a mini symposium. This mini symposium is aptly called as Research Advances in NIBMG. So with that introduction, I would like to invite Dr. Salilan Kurklan. She is a postdoctoral fellow, and she will talk to us about single cell RNA-seq and describe its role in affecting cellular heterogeneity. So please bear with me. And I will transfer it all to Vikram, and he will control once our slides are ready. Okay, just give us a couple of minutes for the presentation.
Yeah.
Thank you.
Anna, are you making ready?
Yes, just give us a minute. He's putting the slides on. It's okay, we don't need to do that. We just need to bring the slides up and share the screen. What is the third one? The third one, what is that? Share desktop. Vikram, sorry, we are having an issue over here. We are not able to share the desktop. Can you send someone over here to do that? Vikram, can you see the screen now? Our apologies, everyone. We have a switch over problem. We are setting it up, but I would just like to say that we have 3 speakers, and we have 2 more speakers after this. Miss Kavya, who's going to talk about 3D organoids, and also after that we have Miss Naina, who's going to talk about the microbiome. So that will kind of bring us to the close of the scientific session, and after that we will move on.
Okay. Kartiki, you can start now.
RNA sequencing, the The stem cells with these different cell types along with the adipocytes constitute the adipose tissue. Blood is also another complex mixture, consists of multiple cell types like the red blood cells, the platelets, the white blood cells like the neutrophils, the basophils, the monocytes. So such cellular heterogeneity within a biological system, precise characterization of cell types and state is masked by using bulk genomic methods. But such challenges can be overcome when using single cell analysis, where like single cell RNA sequencing, where with single cell RNA sequencing, you can profile the transcriptome of each and every individual cells. As each cell type has distinct expression profile, thus revealing the heterogeneity and subpopulation expression variability of thousands of cells. But with bulk methods, you will be able to get only the average gene expression profile from all cells, thus masking the cellular heterogeneity. So dissecting the cellular differences is critical in understanding the development and disease progression. There are multiple approaches for single-cell sequencing, of which we adopted the Chromium method from 10x Genomics. The key to this technology is the ability to generate large number of gems, which can separate the transcriptome of each and every individual cells. Now, what are these gems? These gems are gel bead in emulsions that were partitioned into tiny droplets surrounded by the oil, and each droplet consists of one cell, single cell, one bead along the RT region in the solution. Now these beads, acts as a delivery system to the solution, delivery system of these oligos to the solution. And each beads consists millions of such oligos. And each oligos constitute of 4 components: the read-1 sequence, that is the sequencing primer used for cluster generation in the sequencer, and the 16 followed by the 16 nucleotide 10x barcode sequence that uses a molecular address for each beads. Then the 10 nucleotides unique molecular identifier sequence. These sequences were used during data processing to accurately quantify the transcript. It distinguished the original transcript from the PCR duplicates. The 30 nucleotide poly-DT sequence used for binding the poly-A-tail of the molecule. Apart from these oligos, there are also additional oligos that can be used if you would like to capture additional features like cell surface protein and guided RNA that lacks poly-A-tail. So how gems are generated? These gems were generated by partitioning thousands of cells into tiny droplets. And this gem generations happen in this Chromium controller machine. Once the microfluidic chips containing the sample, the bead, and the oil were loaded into microfluidic were loaded into the chromium controller machine. The solution starts flowing throughout the channel in the microfluidic chips and combines sequentially, where the cell and the enzyme flows from the channel perpendicular to the main channel meets the bead flows from the main channels. These mixtures along the way meets the oil and the aqueous solution then were partitioned into tiny droplets surrounded by the oil containing one bead, one cell, and this is called gems. The gems after generated were then collected, followed by cDNA synthesis. After cDNA synthesis is complete, the gems were broken using recovery reagents, and the synthesized cDNA were released into the solution. The pooled fractions were then recovered, followed by cDNA amplification and library construction. The resulting 10x barcoded libraries were then sequenced, generating transcript of thousands of cells. So the massive transcript profiling of thousands of cells could help in investigating the human disease, tissue development, etc. It is well known that tumors are a complex ecosystem consisting of diverse cell types, including the cancer cells, the stromal cells, and the immune cells. So each cells exhibit distinct genomic and epigenomic profile, hence phenotypic states. So due to such heterogeneity within the tumor regulates tumor growth, response to therapy, metastasis, and recurrence. Also influence interpatient variability in survival period. Although studies have reported that intratumor heterogeneity in head and neck cancer, including oral cancer, and their clinical implication, but multiple of such studies were based on bulk genomic methods, hence masking the cellular heterogeneity of a tumor. We therefore aim to excavate the individual tumors from OSCGB patients at a single cell level to identify their cell type and cell state heterogeneities in OSCGB. And our objective is to examine the cellular diversity in OSCGB by RNA sequencing of single cells and to identify the dominant cell types and cell states in OSCGB. For this, we generated single-cell RNA sequencing profile for primary tumors by profiling 5,000 of such cells from primary tumor using single-cell RNA sequencing, where after obtaining fresh tumor tissues, this tissue were dissociated into single cells using tumor dissociator and tumor dissociation kit from Miltenyi Biotech. After obtaining single cells in single-cell suspension, these cells were assessed for their viability and cell counting was performed. The cells with more than 70% viability were considered for further processing, of which 5,000 of such cells were taken for barcoding and single-cell library construction. The generated single-cell RNA sequencing libraries were then sequenced using NovaSeq 6000. The sequencing data were then analyzed using cell ranges suite from 10x Genomics, where the BCL files are converted to FASTQ files and the .cloop files, which is the output files, were generated and then visualized in Cell Loop browser. This is also software from 10x Genomics. The cluster thus obtained were annotated using the expression of cell type specific gene markers, where cells are classified as epithelial cells, T cell, B cell, dendritic cells, macrophage, NK cells. And each cell shows distinct expression profile along with their expression, gene expression marks, cell type specific gene expression markers. The cell types were also further classified to subcell types. For example, the T cells are further classified into CD4 positive T cell and CD8 positive T cells. Each subcell type shows distinct expression pattern. Like the CD8 positive T cells shows higher expression of cytotoxic associated genes like the granenzyme, the perforin, NKG7, GNLY, also gene involved in T cell activation like interferon gamma, indicating the T cell activation state in OSCGB. Whereas the CD4 positive T cells shows high expression of Treg marker genes and genes involved in T cell dysfunction, indicating the T cell exhaustion program in OSCGB. The coexistence of both the expression state, the activation state, and the exhaustion state could influence treatment response. The B cells were further classified into activated B cells and naive B cells, where overexpression of B cell activation markers including CD38 was observed in activated B cells, and CD20 was found to be overexpressed in the naive B cells. Higher expression of CD38 was reported to be associated with poor prognosis, and higher expression of CD20 was reported to be associated with good prognosis in chronic. The dendritic cells were further classified into plasmacytoid dendritic cells and monocytic derived dendritic cells. And these plasmacytoid derived dendritic cells were known to contribute to the immunosuppressive microenvironment in the tumor. So the presence of such cells could contribute to immunosuppressive tumor microenvironment in the OSCGB tumor. So the cell type and cell expression state heterogeneities was observed within the same tumor. This could influence treatment response and prognosis in OSCGB tumors. Recent studies from the ICGC groups have reported the significantly and frequently altered gene that is specific to OSCGB, including ARID2, MLL4, and other genes. ARID2 is a subunit of chromatin remodeling complex PBAF, which involves in chromatin remodeling and regulation of transcription. Studies have reported the deficiency in this chromatin remodeling complex enhances anti-tumor immune response, where in activation of PBAF complex, genes including ARID2, BRD7, PBRM1 sensitize the mouse melanoma cell to T-cell mediated cytotoxicity. And such cells shows enhanced secretion of chemokines that recruit affected T cells. ARID2 expression was also reported to be negatively correlated with T-cell cytotoxicity markers in many human cancer types. Metastatic renal cancer patients with PBRM loss of function mutation shows improved clinical response to anti-PD-1 or anti-PD-L1. So PBAF inactivation or a particular ARID2 inactivation in enhanced anti-tumor immune response in OSCGB is not yet reported. We therefore characterize ARID2 expression state in OSCGB. Here is the T-SNE plot of the ARID2 low expressing cells and ARID2 high expressing cells among the malignant population and non-malignant cells population. Among the malignant cells population, 42% of the malignant cells shows high expression of ARID2 depicted in red color, and 58% of malignant cells shows low expression of ARID2 that is depicted in green color. So among the malignant cells, almost equal proportion of ARID2 high expressing and ARID2 low expressing cells. But among the non-malignant cell population, higher proportion of ARID2 low expressing cells than the ARID2 high expressing cells. ARID2 was one of the genes that were reported to involve in cancer immunotherapy resistance. So the presence of such population could contribute to cancer immunotherapy resistance in OSCGB tumors. Among the differentially expressed genes by ARID2 low expressing and ARID2 high expressing malignant cells, ARID2 low expressing malignant cells shows a higher expression of cytokines and chemokine genes that could recruit affected T cells, and also genes that involve in chemokinases like S100A7 and CSF3. Also, high expression of gene involved in T cell activation like IL-1A, ICAM1, TNF was highly overexpressed by these ARID2 low expressing malignant cells. The ARID2 high expressing malignant cells, on the other hand, shows higher expression of genes like SDC1 that were reported to inhibit chemokinases. Among the other significantly upregulated genes in the ARID2 high expressing malignant cells like UBE37 and CDK. So these genes were not yet reported to be involved in enhancing anti-tumor immune response. So the genes that are known to involved in enhancing anti-tumor immune response is significantly upregulated by the ARID2 low expressing malignant cells, but not by the ARID2 high expressing malignant cells. So in conclusion, Cellular diversity was observed in OSCCGB that are characterized by different cell types with distinct expression states. And increased expression of cytokines and chemokines by these erythro low expressing malignant cells indicates that these cells could attract diverse immune cells to enhance anti-tumor immune response in OSCCGB. And characterization of erythro expression indicates that there is a link between erythroid activation and enhanced anti-tumor immune response in OSCCGB that may aid in therapeutic implications. I would like to acknowledge the Department of Biotechnology, Government of India for funding my research associateship and System Medicine Cluster, as this study is part of System Medicine Cluster study. Also, I would like to thank the Core Technology Research Initiative, National Institute of Biomedical Genomics. And behind this works is our team. I would like to thank each and every one of them for the contribution. Thank you.
Thank you, Silverine. That was an excellent talk. I'm sure a lot of us who are uninitiated in the single cell field gained much from that, and how we have the platforms and how we can use our employ our research questions and get answers in terms of cellular heterogeneity. So now we move on to, we know often require that we need great model systems, right, to really address complex biological phenomena that are occurring, as Silverine has already pointed out, in the heterogeneity, in both the normal and disease state. This not only improves our knowledge to a great, and depth of knowledge in this field is improved. So what we are going to see in Ms. Kavya's talk is she will describe an organoid system which she is utilizing to understand and gain a depth of knowledge in stem cell biology. So Kavya, please take over. Give us a minute to pull up her slides.
Unmute. একটু ইয়েকে জুম করে দাও তো প্রেজেন্টার জুম করো। ঠিক আছে স্যার, জুম করো প্রেজেন্টারকে। তুমি মিনিমাইজ করো না, দেখতে পাচ্ছ না তো তুমি? আমি দেখতে পাচ্ছি স্যার। হ্যাঁ, তো মিনিমাইজ করে দেখো, এসকেপ করো, এসকেপ। হ্যাঁ, এবার ওকে আগে দেখে নাও একটু। হচ্ছে না না? হ্যাঁ, এবার জুম আউট হয়ে গেল, জুম ইন করো, আরো।
Vikram, can you hear us?
Yeah, I can hear you.
Okay.
थोड़ा একটু জুম করো না, আর একটু জুম, আরো। একটু লেফটে নাও, লেফটে। No, right, sorry, right. উল্টো দিকটা। বাস। Okay. So, এবার স্লাইড শেয়ার করো। স্লাইড শেয়ার করো। Full screen.
Vikram, can you see the slide?
Now we can start.
Okay, thank you.
Hi everyone, it's really a pleasure to be here and presenting as part of Foundation Day lecture. So today I'm going to talk about organoids towards understanding the depth of biology. First of all, we all know that as early as 1855 that a cell is the fundamental unit of life. We all started our journey as a single cell which multiplied and gave rise to ball of cells, further gave to different diverse cell types and organ system which made up an entire individual. This fascinating process is orchestrated at many cellular and molecular levels during normal development and disease, and many crucial intricacies of how it is maintained is still a big question. So Cilirin has beautifully explained us about single cells. Now I'm going to take you all to the organizations of these cells at the tissue level, and which I'm going to explain using a buccal oral mucosal tissue. So I'll explain with a simplified cartoon of this histology section of buccal oral mucosa. As you see here, the epithelial cells that make up the lining of multiple organs are separated from the connective tissue by a basement membrane, and they're further arranged in a way that the stem cells are arranged in the basal layer, which further give rise to the differentiating layers, and finally become keratinized layers. And these cells are separated from the connective tissue. As I said, the connective tissue is composed of collagens and laminins and consists of structural cells such as fibroblasts and capillaries, providing the blood supply to the epithelium, overall giving a structural support to the epithelial tissues. So understanding this tissue architecture is important in modeling them in the lab. So far the model systems that are available are either in vitro 2D cell cultures where the cells are grown on a plastic in a sheet-like manner, or the live animal models in vivo. And bridging the gap between these two, now comes the 3D organoid models, which are self-organizing miniature structures which can be grown in vitro. These are really act as a model bridging between the 2D cell cultures and animal models, because the complexities that I just showed you in the tissue architecture are not captured well either by the 2D cell cultures and are too complex to study in the animal models. So the 3D organoid cultures, with their ease of experimental handling, the cost and ease of live cell imaging, and being physiologically relevant to human in vivo system and personalization, and for their ease in genetic modifications, are exceedingly becoming popular these days. So these, before going into deep about the 3D organoids, we have to understand why maintaining the 3D architecture is so important. So I'm going to explain with example of the work done by Professor Meena Bissell in University of California. So using breast mammary gland as an example. So as you see here, this is the human breast, which is composed of multiple lobules and ducts, and these lobules, these grape-like structures here, are the functional units of the mammary gland, which are further composed of this is the histology section, which are composed of acini-like structures. These are called the acinus, the round-shaped structures that you see here. Now, each acinus is composed of an inner luminal epithelial cells, which are arranged in a circular fashion, and which are covered by outer extracellular matrix and epithelial cells, myoepithelial cells. The luminal epithelial cells during pregnancy and lactation phase produce milk into the lumen, which comes out when the baby suckles. So this is the basic biology of the functional unit of mammary gland. Using this structure, when Professor Bissell had dissociated these cells and grown them on plastic versus when she grown them as 3D structures that maintained this native organization in basement membrane matrix, she interestingly found that cells cultured on plastic completely forgot their hallmark of producing milk. As you can see that beta-catenin is completely absent in cells cultured on plastic, whereas cells cultured on collagen as 3D structure retained their milk producing ability. So she beautifully showed this the organization of this SNI or the mammary gland units in 3D, and this is her talking about her research. This is such wonderful work which describes why maintaining the 3D spatial organization is so crucial in such studies. Having understood that 3D organization is crucial, Professor Hans Klever's group from Netherlands, who works on intestinal stem cells, basically the colonic intestinal cells, where you can see this is a colon overview of the colon, cross-sectional overview. So as you can see here, the colon is composed of crypt-like structures and villus-like structures, where the crypt-like structures has the stem-like, the stem cells, the adult stem cells, which proliferate and gives rise to the differentiating cells, which with time go up and perform their function, and with time slow off after the function is done. So this is the basic structure of a crypt, colonic crypt in intestine. So Professor Hans Klevers had shown that the cells, the LGR positive stem cells from the crypts can give rise to the self-organizing mini gut-like structures when they are cultured in 3D conditions with defined media. So these crypt-like, these 3D mini gut-like structures are very interesting because they also have shown with in vivo resemblance that they have both crypt domains and the villus domains, and also contains lumen spaces which exactly replicate the in vivo morphology of the crypt-like colonic villi. So these structures, this is how they look in culture. With you can see the colonic crypts and means the crypts and also the lumen in between being clear. And further, they have shown that these LGR positive stem cells, which are colored green here, are located in the crypts just like their in vivo counterparts. So this study is really the basis for the modern organoid technology, and has provided basis for people using organoids as in vitro cultures very often. So the ways in which the organoid cultures or the organotypic cultures can be made is either from a whole organ or a whole tissue or a whole slice, where we can transplant them in culture and can be grown, or as a tissue organoids, where the small tissues of the epithelium can be taken and embedded in 3D and grown as an organoid, or like I've just discussed, from stem cells after digesting and sorting single cells and grown as organoids, or also from primary cells after isolating the primary cells and growing them as organoids. There are multiple ways and multiple cellular inputs that we can give in culturing the organoids. So not only from the normal tissue but also from tumors, there are reports of multiple reports of tumor organoids. So this is one very initial study that had shown that in the colon cancer, as you can see clearly from 0R to 36R, the cells in the defined 3D conditions had formed smooth sphere-like structures and formed tumor organoids. And these tumor organoids are similar to the original tumor from which they are derived, and maintain morphologies and physiological functions similar to the original tumor. Such tumor organoids are available in multiple solid tumors such as ovarian, breast, and lung. And further, the organoid cultures currently are being derived from patients with advanced stage diseases and are being used to tailor the needs of the patients in personalized therapies. So having said all this, now I'm going to give glimpses of work that we have done in our lab using the 3D spheroids. So we have collected oral cancer surgical specimens and processed them in lab by finely mincing and enzymatically partially digesting and plating them on collagen coated dishes and growth media. So with this method, we were able to grow continuous cultures of 2 epithelial cultures and 20 cancer-associated fibroblast lines, and we have standardized their 3D spheroid growth in culture using basement membrane matrix. This methodology is published in a recent study, and the cells, I am going to show you the video of the live cell imaging of the 3D spheroid formation in culture. So as you can see here, follow one cell. It grows in size. This is a time span of 48 hours, and you can see the 3D spheroid growing in size. There are multiple spheroids there, but you can see the cell clearly multiplying and dividing and becoming a bigger sphere. Further, this is a video of 3D reconstruction of this Z-stack profiles, showing clearly the 3D organization of the spheroid. This work is done by my lab mate Subhashish Ghosh. And showing that these 3D spheroids are enriched in stemness, we have done gene expression microarray between 2D cultures and 3D spheroids, and clearly it was shown that the 3D spheroids are enriched with developmental gene signatures in the gene set enrichment analysis. Further, the qPCR also showed, as you can see here, compared to the adherent cultures, the organoid cultures are highly expressing the SOX2, LDH, and Notch pathway signals, which are all involved in cancer stemness. And using genetic modification techniques, we are showing here that these genetically modified cells in the spheroid cultures, the green color that you are seeing here is a Notch active overexpression clone, which is active in Notch signaling. And so it clearly shows the upregulation of all the stemness markers and the Notch related pathway genes. Though these cells, when grown in 3D spheroid condition, has shown inhibition to the cancer stem cell specific drug salinomycin, but not to the cisplatin. Using these models, these spheroid models, we have screened novel compounds, novel anti-cancer compounds in collaboration with Dr. Arindam Mukherjee from IISER Kolkata, and this study is published recently. Now coming to the 3D heterotypic cell studies, for enabling the heterotypic cell studies, our previous paper had shown that there are 2 types of cancer-associated fibroblasts that are present, either C1 or C2 type, which differently modulate the functions of the tumor cells. Now we have updated to the 3D conditions. You can see these beautiful scaffolds of where the fibroblasts are making scaffold-like structures and the tumor spheroids growing in these structures. So this we have updated, and then we are really excited about this data. And now giving glimpse of how the organoids can be used in personalized patient care. The recent research tells that organoids can be derived from the normal and the tumor cells and made into organoids, either the healthy organoid or tumor organoid, and can be screened for multiple drugs at a time and for genomic studies, which aids in personalized medicine of the patient, tailoring the needs of patient, and then select drugs that have worked best for the patient based on drug toxicity and then the liver toxicity assays. There are multiple such reports, not only in the advanced cancers such as this paper reporting locally advanced rectal cancer, but also in tailoring the needs of patients with other complex diseases such as cystic fibrosis. And these organoids, like currently we are living through a pandemic, and it's not fair if we don't talk about it. The organoids are also being harnessed in SARS-CoV-2 research currently. That the recent science paper which tells about SARS-CoV-2 productively infects normal gut enterocytes. As you can see here, this is the picture of the gut organoid which is infected with SARS-CoV-2 24 hours. This is the capsid, viral capsid protein, and at 24 hours you can see it's in only one cell, but within 60 hours it is spread across all the cells in the lining of the organoid. And these authors clearly tells that the SARS-CoV-2 readily replicates in abundant cell type of the gut, which is the enterocyte, resulting in production of large amounts of infective virus particles. There are many such papers that are using organoids currently in the COVID research. And interestingly, I have found that it seems the ACE2 expression, the viral receptor, is expressed in the intestinal epithelial cells, and single cell sequencing analysis further suggests that actually the gut lining has higher expression of ACE2 even compared to the lung cells. So these organoids are really helping in proving this, and which tells a critical role of why 3D structures are very important in research. And in a very innovative way of using organoids, again, Professor Cleaver's group has shown that that snake venom gland organoids can be produced, which can be used for the novel therapeutic approaches of dealing with snake bites. And they have shown that these organoids, the snake venom organoids, produce functional toxins that are similar to the wild type toxins present in the genome. And this is really a very innovative way of using organoids. So with that, I would like to conclude that organoids are self-organizing units that recapitulates the key phenotypic and molecular functions of the organ in question. They're very versatile systems to study organogenesis, normal homeostasis, and disease biology. But of course, like with any model system, there is limitation for the organoids as well. They have limited capacity to reproduce the complexity and dynamic environment of the developing organ, because it needs the from the signals from the microenvironment. This is an example of a brain organoid, which is being shown here with the complex structure of trying to recapitulate complex structure of brain. So the future directions are that like organs on a chip. Right now, researchers are working towards organoids on a chip, which is a fabricated cell culture device on which the organoids can be grown. But that's for another day. So with that, I really thank my professor, Dr. Sandeep Singh, for supervising me in this work, and director, Sir Professor Soumitra Das, and PPM Sir Sharmila Ma'am for providing excellent infrastructure and positive work environment in NIBMG, and Dr. Kartiki Desai, Dr. Alpana Dave, Dr. Vikram Roy and team in NIBMG for organizing this event, and the clinical collaborators and all our collaborators who helped us in our research, and all my lab mates. I really thank you. Thank you very much.
We'll come to the next talk of our session. So considering that the number of microorganisms far exceed the number of cells in our body, clearly they have very important effects on the host. So our third talk explores how these interactions with them affect either the initiation or establishment or phenotype, etc. of chronic diseases. So I now invite Ms. Naina Kumari to describe her efforts in using the microbiome research to understand disease. Naina? Okay, I think Naina is in the other room, so
Yes, Naina will speak from other room. Just a minute.
Video on it.
Video Hello. Hello. Hi, bolo. Can you hear me? Hello. Now you can start.
Good evening, everyone. I am Naina Kumari, and I am working as data analyst in the host microbiome interaction lab of Dr. Souvik Mukherjee. And I am very grateful that I have been given this opportunity to speak in this auspicious occasion to represent our lab.
So we are working on microbiome. So first we should know what is microbiome. So microbiome is the ecological community of the microorganisms that coexist with us, and lately they have been ignored as the determinants of health and disease. So the term microbiome was first coined by Joshua Lederberg, and during his time it was known that the number of cells in our human body is 90% of the cells in human body are those that of the microorganisms. So that led to the foundation of the Human Microbiome Project. But recently, it has been contested in the year 2016 that human cell is to microbes is 1:2. So there are as many the human body harbors as many microbial cells as there are human cells in our body. So it is very important that we study the second genome that is of us, that is the microbiome. So the microbiome studies can be performed by two types of methods, that is 16S RNA gene sequencing method and the whole genome shotgun sequencing method. So 16S rRNA gene sequencing, by using this we can classify the bacterial taxa up to the genus level. But by using the whole genome shotgun sequencing method, we can classify not only the bacterial taxa but other microorganisms such as the viruses and fungi, and classify them up to the species and strain levels. Other than that, we can also classify the gene families and microbial pathway can be identified from them. So it is very, as we should know that the healthy microbiome is not same for all. Microbiome is not same for all, and this has been published in a paper in 2014, and they have compared the microbiome of the gut microbiome of the African tribal populations with the Western populations. So in that they have found that the African tribal populations consist mostly of the bacteria, that is Prevotella. It is the mark of African gut healthy microbiome, and which is almost absent in the Western populations. And whereas Western populations are marked by the bacteria, Faecalibacterium, that is the blue ones. And also when we see among the among the African populations, there are also variations from one population to other when we move. So it is important that we study the healthy gut microbiome across different geographic populations, so that we can create a baseline and estimate the differences from the other chronic diseases. The microbiome is not present only in our gut, but it is also present variedly in all our body sites, such as the skin, nasal cavity, and all. And it is like the lower body mass places, areas in our body like skin and nasal cavity should also be studied because these areas also harbor microorganisms and they are important for health and disease. So the healthy skin microbiome has been well characterized in the Western populations, but it has not been studied in any Indian or Asian population. So we sought to characterize the skin microbiome of healthy Indians. In this study, we tried to answer two research questions: that how much does the profile of skin microbiome differ among the healthy Indians, and what are the major differences among them, and what are the individual variation in skin microbiome. So in this study, we have selected samples from 30 healthy female volunteers and of mean age of 29 years. The samples were collected from two regions, that is the forehead and cheek, and the sebum and hydration levels were measured from these samples, and the 16S rRNA gene sequencing was performed, and analysis was performed to obtain the taxonomic classification and phylogeny reconstruction. From this study, we have got major insights into the healthy skin microbiome. So we have found that the sebum and the hydration levels can be a predictor of the skin microbiome. Each unit change in the sebum and the hydration level can cause change in the healthy skin microbiome. So, we have also compared the healthy skin microbiome of our study with the European study, that is UN et al. Published in 2012, and we found that Corynebacterium, which is considered to be a commensal bacterium, and it detoxifies sweat, found to be abundantly present in the European population. But the Streptococcus, which is generally considered as a harmful organism, was found to be abundant in the Indian population rather than not in the European populations. So our study was the first skin microbiome study that has been done on any Asian or Indian population, and it has received 39 citations till date. So since we have profiled the healthy skin microbiome, so now we have entered into studying the chronic inflammatory skin diseases such as the atopic dermatitis and psoriasis, and so that we can find what is the self trigger. And we are also studying into diabetic foot ulcer and maternal and child health microbiome, and we are collaborating with major research institutes like THSTI and SAS, and also with other medical hospitals like JNM and CNMC. So as I said, that the atopic dermatitis is a chronic autoimmune skin disorder, and it is associated with impaired skin barrier permeability due to the dysfunction of the filaggrin gene. So loss of function mutation in the filaggrin gene is found to be associated with AD in different parts of the world. So globally, there is a lack of host microbiome interaction studies in atopic dermatitis. And so we have carried out this study. And in this study, we have selected some stringent exclusion and inclusion criteria, and we have also followed the Williams criteria, so to select the study participants. And in this study, we have selected biospecimens from 8 cases and controls in the ratio of 1:3. And the lesional and non-lesional swabs are collected for performing the metagenomic study. Human blood samples were collected to perform the host genomic study, and human serum samples were collected for performing the cytokine expression assays. So in this study, we have found that Staphylococcus aureus to be highly abundant in the cases, and Staphylococcus hominis is highly abundant in the controls. And it has been reported in other studies that Staphylococcus hominis releases some kinds of proteases that kill Staphylococcus aureus. Other than that, we have found that the fungal, that is a commensal fungi, that is Malassezia globosa, and the commensal bacterium, that is Propionibacterium acne, is greatly depleted in the cases. So as we have performed the whole genome shotgun sequencing, so we have identified microbial pathways of these associated with these bacteria, and we have identified a total number of 182 pathways. Among them, 13 pathways were found to be discriminantly present in the cases, such as the L-histidine metabolism pathway, the arginine and proline metabolism pathway, and the S. aureus infection pathway. So we have found that the genes involved in the S. aureus infection pathway are greatly present, highly abundant in the cases, and these genes inhibit the complement system, and they infer resistance to the antimicrobial peptides. They create B cell proliferation, apoptosis, and thus causes host inflammation. And as I have said, that histidine metabolism is also significantly higher in the cases. So this pathway is involved in ammonia production and increases the skin pH. So L-histidine is one of the component of the filaggrin gene, and it is broken down by the enzyme histidase to form urocanate. So histidase, the urocanate, is a molecule that protects the skin from the UV rays. And when there is an invasion from opportunistic pathogens, or such as the Staphylococcus aureus, the urocanase enzyme is produced by those bacteria, and the urocanate is degraded. So our skin gets exposed to the UV rays and thus cause inflammation of the skin. So we have also performed the host metagenomic study, and we have sequenced the filaggrin gene, and we have identified missense mutations. So out of all the filaggrin missense mutations that is found in and around the world, we have found 5 missense mutations that are found to be potentially damaging as predicted by the variant effector predictor tool. And these 5 SNPs, the damaging allele dosages of these 5 SNPs were calculated and correlated with the microbiome. So we have found that Staphylococcus aureus was significantly negatively correlated with the increasing allele dosage. So this shows that there is a role of microbiome. And we have recently submitted our paper, and it is under minor revision, and we hopefully think that this paper will soon get published. So, we have completed 4 years of our host microbiome interaction lab, and we have received successful funding from DST, DBT, and industrial partners like Unilever R&D. And we have published 2 book chapters and 3 papers, and some are under revision and some are under to be submitted. So, Professor Parthapi Majumdar is the founder of NIBMG and our major collaborator, and we have collaborated with other skin department and of the hospitals in and around Kolkata. So this slide shows that the extensive work that we have performed in our work and our lab members. The extensive work have been performed that starting from sample collection and microbiome DNA isolation, that we have performed it for more than 500 samples. 6-nest sequence analysis have been performed for more than 600 samples, and shotgun metagenomics analysis have been performed for around 150 samples. We have also recently organized an India EMBO symposium on human microbiome, and it is one of the international symposium that was held just before the COVID era, and it was a great success. So moving on to the future scope of a lab, we would like to create and we would like to make a microbiome facility in NIBMG encompassing microbial DNA or RNA isolation from different types of biospecimens. Then we would like to study the anaerobic cultivation of viable but non-culturable microorganisms and host microbiome interactions. Studying the host microbiome interactions by in vitro co-culture method, studying the immunomodulatory effect of human microbiome by metatranscriptomics and metaproteomics approach. And recently, our lab is also a part of the CSIR initiative to profile the Indian gut microbiome. And we have also collaborated with CSIR CDRI Lucknow for understanding the functional role of the significant taxa in development of the antimicrobial resistance in animal models of diabetic infections. And we have done collaborative ventures with the CSIR IMTech for studying the interactions between the host microbiome for immunological studies. So lastly, I would like to sincerely acknowledge Professor Soumitra Das, Director NIBMG, Professor Parthapi Majumdar, Founder of NIBMG, my PI Dr. Sovik Mukherjee and Sonko Nath, our lab member, and all the faculty members and all NIBMG staffs for giving me this opportunity. Thank you.
Thank you, Naina. I hope everyone can hear me.
Thank you.
Thank you. So, very good. Yeah. Naina, can you unmute your mic on that end so that I'll be clear?
Now we can start, Tamamno.
So, thank you very much. This concludes our science session for today. And I congratulate all the ladies for giving such excellent talks, and a special acknowledgement from my side for doing such great work and doing a contribution to science. So if there was really no pandemic right now, and this was not the new normal, we would have certainly have had a cultural blast following our scientific session. But just to be safe, we have restricted ourselves and controlled our urge to have a cultural program in NIBMG. However, I will call upon Mr. Tamagno Chatterjee to sing a song to conclude our program. It's just a demonstration of the tip of the iceberg of our cultural capabilities at NIBMG. So, Mr. Tamagno, if you can.
উড়িয়ে ধ্বজা অভ্রভেদী রথে ওই যে তিনি ওই যে বাহির পথে উড়িয়ে ধ্বজা অভ্রভেদী রথে ওই যে তিনি ওই যে বাহির পথে আয়ের ছুটে টানতে হবে রশি ঘরের কোণে রইলি কোথায় বসি ভিড়ের মধ্যে ঝাঁপিয়ে পড়ে গিয়ে ঠাই করে তুই নেড়ে কোনো মতে উড়িয়ে ধ্বজায় অভ্রভেদী রথে ওই যে তিনি ওই যে বাহির পথে কোথায় কি তোর আছে ঘরের কাজ সেসব কথা ভুলতে হবে আজ কোথায় কি তোর আছে ঘরের কাজ সেসব কথা ভুলতে হবে আজ টান রে দিয়ে সকল চিত্ত কায়া টান রে ছেড়ে তুচ্ছ প্রাণের মায়া চল রে টেনে আলোয় অন্ধকারে নগর গ্রামে অরণ্যে পর্বতে উড়িয়ে ধ্বজা অভ্রভেদী রথে ওই যে তিনি ওই যে বাহির পথে ওই যে চাকা ঘুরছে রে ঝনঝনি বুকের মাঝে শুনছ কি সেই ধ্বনি ওই যে চাকা ঘুরছে রে ঝনঝনি বুকের মাঝে শুনছ কি সেই ধ্বনি রক্তে তোমার দুলছে নাকি প্রাণ গাইছে না মোর মরণ জয়ী গান আকাঙ্ক্ষা তোর বন্যা বেগের মতো ছুটছে নাকি বিপুল ভবিষ্যতে উড়িয়ে ধ্বজা অভ্রভেদী রথে ওই যে তিনি ওই যে বাহির পথে
Thank you, Tamogni, very, very much. That was excellent, and that concludes our program for today. But before we go, it is my pleasant duty on the behalf of the organizers, Dr. Arindam Mehta, Dr. Nalabu Basu, and myself to give the vote of thanks. I start with thanking our director, Professor Soumitra Das, for all his support that he has given us in organizing this event, and thanking Professor Partho Mazumdar for all his efforts in getting the speaker, Professor Sunetra Gupta, and we thank her too for both of them for taking time from their very busy schedules in making this event a success. So I thank them. Next, I would thank Mr. Bikram Roy and his team, because this entire event was based on an online version of it, and it went very, very smoothly. And I really thank all of them for making this a big success. I thank the other organizers which were part of our group was Dr. Sandeep Singh and Dr. Molinath Acharya, as well as Mr. Nabarun Mukherjee, who took care of other logistics that were required to make this event a success. I thank particularly Ms. Alpana Dave, who has also been on our side and has done countless emails, and particularly I thank her for design of the flyer. I will thank the students. And I particularly wanted to mention this is the first time I think on a foundation day we have had 3 students speak and describe their talks and how they're advancing the field of genomics. So I thank all 3 of them for giving excellent talks, and that was also on a very short notice actually. And we wish them all the success on our part so that they do very well in science and become beacons in their own careers. Okay. Last but not the least, we thank all of you for taking time out from your busy schedules and joining our online program and helping us celebrate the 11th Foundation Day of NIBMG. I request all of you to kindly join us for refreshments at the main door of NIBMG. And with that, we will conclude this evening. And good evening and see you again. Bye.
Kartiki, I'm closing this session, huh?
Yes, please. Thank you.
Yeah.